Hello and a very warm welcome to everyone who has joined us today. My name is John Norman. I am Head of Marketing here at Optibrium and moderator for this webinar. As always, it's great to see so many people signing up for our webinars, and today we will be discussing harnessing generative chemistry and exploring how scientists can guide compound idea generation. And for this presentation, I'm joined by Ed Champness, CSO at Optibrium, Peter Hunt, who is Optibrium's Director of Computational Chemistry, and I'm also delighted to welcome our special guest, Joe McDonald. And here's a quick overview of today's agenda. We will begin with our presenters engaging in a roundtable discussion to dig down into the challenges of generative chemistry, the changing landscape, and the latest advances that are being made. And with Joe being in the US, Peter on the south coast of the UK, and Ed in Scotland, we will be bouncing this conversation around the globe. We will then follow the roundtable with an introduction and demonstration of Inspira, the latest technology being developed as part of the Stardrop modules kit. Then finally, we will open the floor for the Q&A session. So now it's time to hand over to our host of presenters, who once again are today's host, Ed Champness, our presenter, Peter Hunt, and our guest, Joe McDonald. Ed, I'll now hand things over to you.
Excellent. Thank you very much, John. So yes, I'd like to start by saying just a couple more words really to welcome today's presenters. And certainly I'm very pleased to firstly welcome Joe McDonald. So I suspect a number of you already know Joe. For those that don't, Joe has extensive experience within the pharma industry. Joe is skilled in comp chem, cheminformatics, bioinformatics, and life sciences, and has a PhD from State University of New York. Joe was, until recently, the Executive Director for Predictive Drug Substance Research at BMS, and prior to that was the Executive Director of comp chem, cheminformatics, and bioinformatics at Celgene. I'm also very pleased to have my colleague Peter Hunt with me today. Peter trained as a synthetic chemist and completed his PhD at Imperial College, London. He moved into comp chem during a postdoc at the 3D Centre in Australia and has since led the CAD groups at Merck and then Novartis, before we were very lucky to have him join Optibrium as Director of Computational Chemistry. So thank you both very much for joining me today. Today we're talking about generative chemistry, and in particular, we're focusing on the compounds that are generated along with their potential qualities, and that's rather than really the synthetic routes by which they might be synthesized. To get us started, I wondered if you could perhaps share a quick overview of your experiences with generative chemistry, Joe.
Sure. So back as part of Avila and Celgene, I had a naive but aspirational goal that we would use enumeration and predictive methods in order to guide chemistry towards desirable target product profile. And the idea was, we would enumerate a collection of compounds, either reaction-based or Marcouche-based, and that would generate a large library of compounds from which chemists could examine that set of compounds to determine whether they were moving in the right direction or not. My observation is that there are a lot of steps in that process. And then once you've completed it, you're confronted with potentially thousands of compounds that you have to sift through in order to determine whether they have the properties of interest. And so the idea around the product that you're going to describe today was sort of born from the idea that NOVA, as a tool that is currently within Stardrop software, has some of the right stuff involved. There are known transformations. But if there's a way that chemists can see the generative process ongoing, and they can interact with it in order to direct the compounds towards the types of transformations that they're interested in, like that interactive process of being guided by human, that was sort of the idea of trying to improve on the original idea of which ultimately was being overloaded with data to try to make it a problem that was more tractable and easy. Peter?
I'd have to say that my industrial experience has been, everybody can generate ideas, but these are one idea after another. And as Joe was describing, the enumeration of much larger libraries of compounds, that was really reaction based. It was the purview of sort of specialist groups who were dealing with combinatorial chemistry or the computational chemistry for the theoretical process, but driven by particular reactions that people could do. And what I see now really is this thought of looking at compounds as individual things within the compound space, this enormous compound space. And it is really something that has been brought more out of the specialist groups, more into the hands of synthetic chemists themselves. And it is really this particular process of how do you take that huge chemical space and be able to manage it. So those were my experiences with the generative side.
So I think actually you've both touched on a couple of things I was interested to ask you next. But Joe, I wondered if you could say sort of anything in particular about sort of key challenges when it comes to generative chemistry.
Well, I've seen more recently an issue where generative models can produce compounds that require a fair amount of book time to try to research how difficult they are to make. And I know that there's a lot of active research in that area, but I thought that this was another attractive feature of NOVA as well, and particularly like the BioStare database that's part of StarDrop, where these are known transformations that have been applied, in other sort of published use cases where we know that there's, a very sort of simple one point change that's associated with the compounds there. So it doesn't get directly to the synthesizability, but at least informs on sort of plausible near transformations from a reference compound. And I find that to be something that chemists can gravitate towards instead of having to have a full retrosynthetic pathway. Peter?
Yeah, I agree wholeheartedly. I mean, I've faced the idea of having to persuade medchem teams, oh, isn't this a wonderful idea? You should be thinking about doing it. And the pushback is very much on the case of, yes, that might be a wonderful idea, but how do I make it? The previous ways of enumerating libraries had the great advantage in the fact that they were based around known chemistries. These were things that chemists knew how to make. They know the reactions, they know the limitations of the reactions. And now that we are exploring the whole of chemical space in here, it now becomes a real problem in persuading chemists to take up these ideas, because there is no direct, obvious route to be able to make them. And with Inspira, as we have now, as Joe described, these are ideas that are generated from known references. These are compounds that are existing, and therefore the changes that we make form nice pathways in terms of being able to say, yes, I know how to make this compound, and we can suggest derivatives of that compound or changes to that compound, which should be perfectly reasonable in terms of either the existing chemistry or small modifications to the existing chemistry to be able to achieve that compound. And therefore, it's a lot easier to be able to think of these compounds and their uptake of these ideas, because they come from positions of knowledge and positions of where you can get to the compounds that are the reference compounds.
Thanks, Peter. Yeah, so I think it's fair to say that everybody is talking about AI these days, and I wondered, Joe, do you think advances we're seeing in AI are changing the generative chemistry landscape?
With absolute certainty, they are. We're seeing increasingly sophisticated approaches of combining the generative chemistry with sort of the use of Bayesian methods and active learning in order to optimize a constellation of properties for the compounds. So I think the use cases for generative chemistry are numerous and hopefully like a golden for our future. But finding ways to make tractable ideas, I think that's the key element.
I have to admit, I'm a little bit more pessimistic of the term AI as a general view, and I think that there's an awful lot with respect to chemistry that comes down to experience, comes down to, yes, I have confidence in being able to make this particular compound because I've had experience in these types of reactions and these types of products before. And I think AI still has a long way to go to try and encapsulate all of that chemistry knowledge and feeling from the lab of being able to know that you'll be able to generate this particular molecule with a reasonable chance of success. I mean, yes, there is always with AI on any machine learning techniques being able to suck up so much more information than one person can ever do. And therefore, it is much more likely that these methods will be able to find ways, find routes to suggest to make some of these compounds, but it still has to be that the chemist has experienced some of these and to have the confidence and the trust in those systems to be able to say, yes, okay, this AI has come up with this particular route. I trust that this is likely to work.
So despite perhaps a little bit of that pessimism, Peter, where do you think it could go? Where could we get to?
Oh, I mean, I definitely agree with Joe in respect to the current research on reaction predictions and the ability to retrosynthetically analyze a particular compound and be able to sort of say, yes, I can make this compound in X number of steps from particular starting materials which are commercially available. I still think that those systems have a way to go, but they are orders of magnitude better than they were when I was last in industry. So I think that even in a short space of time, it has improved immensely. The next challenge is to actually get those types of systems into a chemist laboratory, into a chemist desktop, and being used routinely, almost perhaps without thinking or without, as a first literature search is, okay, I have a particular idea. How would this methodology suggest to predict it, and what are the references that I can get for those? As soon as that tool becomes available, then I think it will be a no-brainer. It'll just be yet another tool like an HPLC that a chemist would use.
Did you have any thoughts on that, Joe, in terms of the potential future?
Yeah, I can add to that. So, I'm sure everyone on the call, and I can explain it in an oversimplified way that, deep learning methods do well on chess because we don't really care exactly what's going on under the hood, as long as ultimately the strategy is a winning one. That's not like laboratory research. So each project is different. Each set of issues that the team is facing is different, and they're not just scientific, there are pragmatic issues in play. And so the human has to be involved. The human has to set the strategy and the path forward. So ideally, you want a system where it ultimately sort of makes human decisions more effective, but it doesn't replace the human. The human still has to guide the project through a nearly infinite gauntlet that can't be known by the machine. And so that's, to me, the secret of sort of moving generative science forward, is allowing the scientist to basically control and interact with the process.
And Peter, did you have any thoughts on that sort of question of the importance of human expert interaction that Joe's sort of brought up there?
Again, I think this is somewhat of a no-brainer, certainly from the computational chemistry side. I say the thought of coming up with ideas as a computational chemist and then having to go to a medicinal chemist and sort of say, look, aren't my ideas wonderful? Well, I am competing against all of the wonderful ideas that come within a medicinal chemistry project. And that fight and that particular way of deciding what gets made next, the humans have to be involved. It cannot just be a case of, oh, the computer says this. I know from experience that that is not going to be the way to actually get these compounds moved forward. The fact that you have the human involved, being able to sort of craft what is there and coming out of the computer, that is something that will move projects forward, not just the picking a molecule out of thin air and saying, yes, of course, we predict this to be wonderful, go away and make it.
Thanks, Peter. Yeah, I mean, I think it's fair to say that these are all topics we've been discussing with Joe actually over a number of years. These actually resulted in our investigating and developing in collaboration with Joe what we're now referring to as Inspira. So to set the scene, I've got a couple of slides here just to describe how we approach the project. But as John mentioned at the beginning, Peter is then shortly going to give a demonstration of what has come out of that collaboration. So the general principles we really focused on were having a system that could constantly generate, evaluate, and optimize compounds in the background. So the idea being that the system could present new ideas to users at their computer as they're doing their work to help them explore a whole variety of optimization strategies. And as Joe and Peter were sort of touching on at the end there, the key to it that we wanted to focus on was capturing and learning as users interact with the system to propose the most relevant ideas that are coming out of the system. So the new ideas need to be influenced by users' objectives or the compounds that they're working with, or perhaps the ideas that the user selects or rejects as they're proposed. And just to give a picture of where we started in our thinking, for those of you familiar with our StarDrop software, this will look very, very familiar because we took the view that we have a system, we've got some software already in which we can automatically generate compounds, far more than one can easily eyeball. So as part of the mock-up, we wanted to think about how we could take advantage of that ability to generate lots of compounds, but present them to the user in a way that allows them to consider more possibilities than a compound or compounds that they're looking at any given moment. And this is very much an early mock-up. This certainly pushed my PowerPoint capabilities. But the principle behind it was that whatever we're looking at, the system would be generating ideas and feeding them into view. Of course, ideally not as fast as this, but you'll probably get a feel for where this idea went as sort of Peter sort of gives a demonstration of the embodiment we came up with. But the principles were very much sort of captured in this early mock-up. Of course, the key to what we wanted to do was prioritize the ideas before they're presented to the user. Inspira can generate huge numbers of those ideas. And so we wanted to make sure that the probability of any idea being displayed related to what the user is interested in, or what the system feels the user is potentially interested in terms of its chemistry, in terms of its sort of chemical diversity and its properties. So the idea is that as the user starts to interact with ideas as they're presented, Inspira can learn from those interactions to infer what their requirements might be in terms of the chemistry and the properties of those compounds. So the underlying engine is essentially learning a user's goals based upon that interaction. i.e., if a user interacts in a positive way, selecting an idea, that's a good thing. The engine's going to learn that that's something they like. Whereas if a user throws an idea away, the system knows that that wasn't something they wanted to see and can take a steer in a different direction. I've hugely trivialized the underlying mathematics which is going on behind the scenes here, but if you think of where the user wants to be in terms of a sort of hotspot on that sort of in that property diversity space, as the user continues to interact with the system, that hotspot is going to move around. Indeed, it's going to do a lot more than that. It's going to change shape. Indeed, it might get broad, it might get narrow. There might be multiple hotspots in that space, but hopefully that gives a feel for the kind of thing we were trying to bring together in terms of properties and diversity of chemistry being learned from the user's interactions. So that was sort of where we set off from. That was what we were shooting at. And as I said, I'm going to hand over to Peter now, give him control, so that he can give you a demonstration of how we've embodied Inspira on the back of the work that we've done.
Thank you very much, Ed. So for this particular demonstration, what I have open is Stardrop with Inspira installed, and it looks very much like you would expect a normal Stardrop project to look like. The aim of this is looking at a data set of a couple of hundred compounds. We've got 200 compounds here, directed against DPP4, so dipeptyl peptidase 4. And back in the development stage of these compounds, there was a worry that some toxicities associated with these compounds were related to their ability to be selective or non-selective against the other isoforms of DPP. And so what we have here is a data set of a couple of hundred compounds from 4 different series, and we have a scoring profile where we have models to predict the activities at different isoforms of DPP. And we've also integrated a few phys-chem properties here, and we have scored all of these compounds and found, as the box plot on the right-hand side shows, that whilst we do have some compounds within these series that have a modicum of likely success, the score is above 0.3 here, we do seem to have quite a number of these compounds that really are not meeting the goal of a selective DPP4 inhibitor. And so what we would really like to have is a way of taking this particular data set and trying to generate ideas driven towards improving the DPP4 selectivity and also keeping some of these good phys-chem properties. And as Ed showed in the mock-up, one of the ways that we can do that is with the idea panel. And he showed in the mock-up that idea panel used to be in the design area, where we were looking at usually individual changes to a molecule. But instead of crowding it into this section, we can bring in the idea panel to the bottom of the table here. Now, as I'm speaking, what I hope you'll notice is each of these panels, the compound in there is an idea. It has been generated based upon the NOVA transformations and the methodologies that we have within NOVA, based upon and in using all of the compounds out of this DPP4 data set. So usually within NOVA, it's a very active process. You have to think, oh, I want to take this molecule and I want to find ideas based upon this reference that take me in a particular direction. Well, the idea here is really saying, well, I'm not terribly interested in exactly which one of these compounds is used as a reference. I just want to find changes and directions that will help me get to a more selective DPP4 compound. And so each one of these compounds is used as its own reference. And the display down the bottom, the idea panel, the property is selective DPP4. This is the column of the scoring profile. So this is what's being used to really evaluate each of these ideas that come through. And there are a pool of ideas being generated, and each of those gets floated to the surface in terms of this idea panel, and I get to see an indication of the properties of these compounds. Now, the first thing to indicate here is that for this idea, I'm looking at the DPP4 inhibition, and I have my histogram and the score associated with it. Now, this at being 0.08, I showed before that we had distributions, means between about 0.1, 0.2. You can see that this is not going in the direction that I really want. Compared to the data set, this is lower than the average. And so you can see that there is a blue border around this particular idea, indicating to me that based upon my particular criteria, this is going towards a lower numeric value. That's not what I want. I want to have things that have higher numeric values. Now, I haven't told Inspira what I want, but I can interact with these ideas as they come through. And so when I hover over any of these panels, you'll notice that there are some indications that come up in the top left, top right hand corner. These are the ways that I can influence the inference engine behind Inspira and say, no, okay, this is an idea, but I'm not really interested in this idea. So I am going to say, no, I dislike this idea. Give me another one. And I can look through again. No, this is poor, I don't want that. This is poor, I don't want that. That's too simple. That's poor, I don't want that. And now I hope you're noticing that as I'm going through just influencing these ideas, I've got a compound here, 0.35. That's interesting. Yes, I like that one. No, that's poor, I don't like that. Don't like that, don't like that. Or that one. Yeah, that one's okay. It has an aldehyde in there, but I don't necessarily think that's doing too much. And essentially what I'm doing in terms of just taking out these particular ideas is biasing the inference engine to be able to give me ideas that are increasing DPP4. I hope you notice now that after having interacted with just this few minutes here, all of these ideas are now with a red border. These are now interesting. And I'm going to like these particular compounds. These are going in the direction that I want to go. Now, looking at this, we've got selectivity that is really quite good. 0.5, 0.7 here. If I want to keep any of these particular compounds in view longer, I can pin it to this, and these cells are now not going to be replaced every 30 seconds or so. If I really like a particular idea and I have perhaps a little soft spot for the cyanopyrrolidines, given my Novartis heritage, I can actually add these compounds to the data set. These are getting to be good ideas. Just unpin that one. And I can add these compounds and sort of say, yes, I'm liking these. I'll consider those a little bit later. So if I just scroll down the table, you'll notice that here are the compounds that I've just been adding to my data set. So I can look at these compounds later on. You may also notice that as I've been liking all of these cyanopyrrolidines, that actually this is the sort of chemotype that's being presented. So not only does the inference engine take account of my preferences here in terms of my likes, my dislikes, what I've added to my data set, that's a really strong like. And it promotes the floating of ideas into the idea panel, which looks similar to the ones that I've given my preference towards. So the liking of compounds gives me more compounds in that particular area of chemistry space. And so not only can you influence what the ultimate value is, the direction that the inference engine takes, but you can also, using this plus and by adding this compound to the data set, really start driving towards suggestions from the pool of ideas that are more in tune with where you want to go. So if you like a particular chemotype, you could start going through here and sort of saying, yes, I like that, and show me more of these type of ideas. If you get to a point where actually, no, I'm fed up with these ideas now, I want to think about something new, then we do have the capability of just saying, right, ignore these selections. And if I ignore a few selections often enough, excuse me, I can possibly get a resetting of the influence that I've made on this inference engine. Now it seems at the moment I'm not getting other ideas other than these cyanopyrrolidines. I've obviously influenced the inference engine enough to really sort of saying, well, that's what you're interested in. Surely, I'll provide you with some more ideas. If you get to this sort of stage, then you can always reset. And the reset function takes that influence that you've had on the inference engine and takes it back to the default. So now you can see that it's coming and projecting ideas from different chemotypes. If I like those, and again, yes, now we're getting into perhaps some different ideas, simpler ones. Now we're getting back into blue borders here. We're looking at DPP4 activities that are going lower. And that's because I've reset the inference engine. I now have to now influence this again and sort of say, no, I'm not interested in these. Well, I have some, again, because I think I've added these compounds to the data set, it's bringing these through. So this is the way one can go through this particular data set. Oh, I definitely don't like that one or that one. And bring your own ideas to the party. But what this doesn't do is try and bias and expect you to come in with that idea right from the start, because Nova can generate ideas based on any of the compounds within a particular data set. I've got a couple of data sets here, so I could be looking at DPP4 selectivity with this one, and then I could go into a different data set and pick a different property. Perhaps I just wanted to have log S, and I can explore ideas based upon that. And each of these individual data sets has its own individual pool of ideas that you can start influencing and seeing how your influences come through in the compounds that are suggested and promoted to the top in the idea panel. So this is a very undirected way. We're not picking a specific compound to look at. This is just taking that whole data set and generating large numbers of ideas. But what we also have within Spira is a more directed approach. And so if I select a particular compound from this data set, you'll notice that in the design tab here, I should just rotate that compound around a little bit more, we have a couple of extra buttons, because there are occasions when you would like to have ideas suggested to you, but you do have a specific compound in mind, or you do have a specific area that you want to investigate with a particular compound. And so what we've provided within Spira is when you select a compound and you see it in the design tab, with these buttons, you can choose. The first one will fragment a molecule into the rings, the chains, the substituents, and you can choose what sort of property you're looking at in terms of what are the things that you would like to influence. And so the first one here is the fragmenter, and it picks automatically a particular ring or a substituent. If you take a mouse and hover over a different area, you can see the first set of suggestions that come through. And very much like the idea panel, these suggestions have a little ring around them to see whether in fact these suggestions are likely to be increasing numeric value or decreasing the numeric value of the property that you're interested in. And at the moment, I'm just flicking through these particular ideas using the middle mouse button, the scroll wheel, and in this, I don't want to increase Herg because I've got the property of Herg here. So I'm going to get rid of that idea. I don't like that. I don't like that one. That one, it's all right, but I'll ignore it for the moment. Oh, okay. I like that one because it's blue. And then I will go through a few more ideas and I can make suggestions, or at least accept. Yes, that one's another idea. Yes, I'll accept that one. And you can move through and go to different areas, be able to say, yes, I like that one because it's sharply blue. It's really going to reduce Herg. I flick through another sets of ideas. That's all right. That isn't. You notice that as soon as I'm getting rid of an idea, it's filled with another member from the pool. Ignore that one because it's an aniline. Certainly ignore that one. Get rid of that, and so on. Now, having selected a few of these ideas, if one hits the blue arrow, then what that fragmenter does is saying, if you're interested in this particular change in this particular part of the molecule, then potentially the combination of those changes will also be interesting to you. And so being able to look at particular areas and make suggestions, selections, as I'm looking through, you can see that these selections are recorded. I've got the ticks there. I like those, and I've made changes at this point, and there are a few suggestions here. It does the combinatorial expansion of those, and having added those to the data set, I've now got a new set of compounds that I can be interested and evaluating. Just one thing about these particular compounds, because I've been looking at the Herg activity, I now have Herg here as a nice high bar, but you can see that the overall DPP4 selectivity, that's not very good at all. So one of those views of being able to say you can design against one particular property, but if you've got others that you're interested in, then we would be looking at a scoring profile as being the way to go. But this is us taking the lead and saying, right, we will split up this molecule very specifically into the rings and the substituents and possibly the chains. But what if you want to come through with your own ideas for here? And this is what the final button that I was going to look at here, where it provides you with the ability to say, right, what I want to do is actually replace the whole of this benzylic unit. And so by the use of this button and selecting a part of the molecule that you want to direct the change, it will now go away and make suggestions for that. And then when it's generated those molecules, I hover over it, there again, it comes up with the same, but now it is for this whole unit, not just the systems that we had before. And so again, I can go through these and scroll through the ideas, and perhaps if I'm interested in a particular idea, I can click on the image. And when I click on the image, it actually makes that suggestion for me. This now becomes a new starting point, a new way of being able to generate some different ideas. So you can go stepwise from a known reference and make further changes, either taking a specific reference here, or perhaps deciding that actually you want to look at the head group end now, and it will generate some more ideas for that. And so here is a way that you can influence the change in a molecule, be able to take the suggestions and they're automatically generated for you without you having to go in and know exactly how to drive things like NOVA and other generative methods, to be able to see these ideas and consider them and be able to bring your influence to what gets shown next. So again, I'm looking for things here with blue borders because these are the ones that are reducing my value of Hg. Now, if I'm interested in those, then again, I can add, in this particular case, it takes the last set of selections I've made. And so I've now selected 3 different head groups and I get to see those compounds in my data set as well. So this is a demonstration of Inspira. This is a rather perhaps artificial environment, but as I finish and hand back over to Ed, then we do actually have a real life example where we've used this underlying technology and we've published on this quite recently. And so with that, Ed, you can show the publication that has come out very recently on the open source malaria initiative. These are compounds that we generated using the underlying methodology to Inspira, evaluating the compounds and suggestions that were coming through, driven towards getting better derivatives from a particular triazolopyridazine core. And this publication covers the success of our designs in this particular open source project. And so with that, thank you very much. And I will hand you back to Ed.
Oh, thank you very much, Peter, for that demonstration. And hopefully that gave you all a good feel for really how we approach the question of putting generative chemistry into the hands of chemists and facilitating that sort of interaction to enable guidance towards particular goals. We do have a number of questions coming in already, but before we open up and sort of address some of those, I wondered, Joe, is there anything you'd like to say, given that you were very much a catalyst for this whole collaboration and this project? Is there anything you'd like to say about really how it all came together by way of a conclusion here?
Yep. So when I initially approached Actibrium with the idea of humans being in the loop and interacting with the system, you know, it was just an idea. And there were a number of inventive steps that needed to transpire in order to ultimately create the application that they're talking about today. So I thought it was a good collaborative process, and I enjoyed interacting with Optibrium as colleagues in developing and testing the concept. Yeah, one of the sort of, I think, hallmarks of the success is they took something that was more or less a simple PowerPoint slide deck, and they turned it into a real product that actually works, and with evidence that it has applicability. And so they made the idea much better than the original concept by turning it into something that was pragmatic and useful. And so I am very much appreciative of the sort of the collaboration that we undertook together. So yeah, I'm definitely an advocate for the collaborative element of working with Optibrium. I'm not being paid for that, by the way.
Thank you very much, Joe.
Yeah, thank you, Joe. As I said, there are a number of questions that have been coming in. So, the first one is, it's easy to generate far more compound ideas than can be synthesized due to time and cost, even when ideas are filtered by synthesizability. Are the current scoring functions able to provide enough enrichment to help prioritize the ideas? I know this was asked before the demo, and the answer is a little subjective, but Peter, do you have any thoughts on that?
Yes, I do, in the sense of I would always be looking at the scoring profile, because we are in an industry that is multi-parametric, and essentially there's always more than one problem that you're trying to solve at one time. And therefore, I think that if you happen to have the ability to predict what a particular new structure is going and how it's going to behave, then you have the ability to drive Nova and to drive Inspira to get you to where you want to go much more quickly. Now, you can have faith that your prediction for an activity is going to be wonderfully precise. These models that I showed for the DPP4 activity, I'm dealing with 4 different models there. Each one of those will have a particular error. However, it is still increasing my chances of success. But because we take that error into account, we can deal that with the multi-parameter scoring and be able to come up with a good rank ordering of compound based on that score and the inherent uncertainty. And so I think that something like the multi-parameter scoring profile is something that will increase your chance of success, given your ability to calculate the properties that you're interested in. And therein lies another story. I would point people towards AutoModeler for that, to be able to generate these sorts of models from a topology of the chemical structure. But that's what you need. You need to be able to have a good estimation as to the properties that you're interested in. If you don't have models for it, well, you are going to be exploring in the dark. And really, there is nothing that will provide you with a magic bullet if you don't have a way of being able to make a prediction that you can rely on.
Thanks, Peter. There's actually quite a load of questions coming in, so I'm going to try and get through them. And just to say, if we don't have time to address them all, we will certainly come back to you with answers. I appreciate these coming through. The next one is a question about asking about the human active learning, and asking, without that, would the algorithm actually be able to reach any goals on its own?
I would say I would say no, because it doesn't know what you want. it will produce a whole set of ideas. It will evaluate those ideas and be able to produce an evaluation, a score, a prediction of some sort. You would then have to look at the pool and then be able to say, okay, actually I'll take that little subset of those ideas because they they produce scores that I want. The whole process of this is that the pool of ideas that are generated is not static. It's something where you will generate ideas, you will interact with it and be able to say, look, now you're going in a direction that I want you to go. That's good. I'm not actively developing, make this change to this molecule. I'm saying you're going in the right way. Provide me with more ideas that are similar to this. And in that way, you can help drive this pool of ideas in the direction that you want. You will get there a lot quicker if you interact with that pool. When I say the pool is not static, as you interact with it, you say, I want to go in this direction. Well, it will add more molecules that are going in that direction. And because the pool is a fixed size, it's about 20,000 per data set, all of the lower scoring ones, well, they'll get drained away. And so because we're not putting because we are putting influence into that pool, it will drive towards the direction you want to go. If you don't interact with it, then you'll have a pretty static pool and it'll just be random chance as to whether it contains a molecule of interest to you.
So Peter, the next question is, what's the source of the synthetic chemistry that the generative engine uses?
Right, well, in NOVA, we have a number of ways of generating new molecules. What we have predominantly as the chemistry transformations, and these are not necessarily actual reactions. These are structural changes that have been made to the molecules that we see and we can apply. So we have two sets, a default set that we ship with NOVA that is fairly standard, simple transformations, changing a phenyl ring into a pyridine, or changing a phenyl into an indole, or an amide into a sulfonamide, things like this, the addition of fluorines, taking away methyl groups, these type of structural changes. But we also have the BioStir database. This has over 27,000 transformations that have been abstracted from the literature that have been used in medchem projects and that have been shown to be beneficial to that project. And so these are a lot more specific. So it could be ring expansions, contractions, changes of cores, a lot greater diversity of transformations, but they all come from the published literature and they've all been used within projects, and they've obviously been beneficial to that project and therefore could be beneficial to you if you happen to come across the same sorts of chemistries in the targets that you're working with. And so essentially these are changes that have been made, they have been beneficial, and therefore they could be beneficial to you in your projects. We do have other data analytics techniques, the match series analysis, looking at the SAR within your data set. We can use that particular procedure to again try and make suggestions for new molecules in various areas. That's a part of Inspira that I didn't have time to demonstrate. But we also have in NOVA reaction-based and scaffold-based library enumerations. And in this, one would have to be selecting particular chemistries. But those are the methods that we have within NOVA, and it's the first two really that are there, the transformations that are used in generating these ideas for Inspira.
The next question is whether or not this could be used in combination with Surflex eSIM 3D.
Yes, and indeed, I mean, we're coming to essentially the evaluation of the worth of an idea. And so if the worth of an idea is that it has to be sterically and electrostatically similar to a known bioactive conformation, then yes, that is something that can be evaluated and that can be used for the new ideas. Now, with version 1 of Inspirer, I don't think that that is available at the moment, but you can consider that not only can you do a ligand-based alignment, you could be doing docking, you could be doing structural-based drug design, and therefore could you involve a docking score in the suitability of a compound? And we all know that, you know, docking studies, depending on how flexible, how rigorous you want to be about producing a good docking score, that can take a long time, and therefore being able to do those dockings alongside the Nova generation, which tends to be quite a fast process, that would require some server communication and really being able to farm those jobs off to evaluate those compounds either in docking runs or in ligand-based alignments. And therefore, that should be something that will be coming in later versions of Inspirer. But yes, it is perfectly possible to think about ways of evaluating the worth of a compound and integrating those evaluations into this idea generation process.
So there was a follow-up to that, and just really the last question I want to touches on what you were saying, Peter. It was, has Inspirer been released? And the answer is not yet, but it is coming soon. So we will be certainly sending out plenty more information. So please watch this space, but it's certainly on its way. I apologize that there are a whole load more great questions, and I'm sorry we haven't had time to address them right now, but we will be back in touch with you to make sure all of your questions are answered. As John mentioned at the beginning, we have an extensive archive where we keep recordings of all our previous webinars, and of course we've been recording this webinar. So please take a look at what we have on the Optibrium community, and this webinar will be up there very shortly. We'll certainly address the questions that have come, but please get in contact with info@optibrium.com if you have further questions. It really leaves me to thank both Peter and Joe for presenting today, and thank you all of us for joining today's webinar. Thank you very much, and goodbye.